Good day, my dear students. For this session, we will be discussing all about the cells, from the different types of cells up to the cell cycle and cell division, and then also the differences between a prokaryotic and a eukaryotic cell. So, before I start my discussion, I would like to share my screen. So please give me a little bit of time to open my tab. And I hope you can all see it. Let me just check if you can really see my slide. Okay, that's it. So again, we will be discussing all about the cells, um, from the definition, the history, and then the conceptualization of the cell theory, the structures and function of the cell, corresponding functions of the structures of a cell, the different movements of the substances in and out of the cell, and then the cell cycle and the two types of cell division. Okay, so what is a cell? Basically, the cell is like the ground, the solid grounds on the study of general biology. It is because the cell is the basic level of structural and functional organization of all living things. So, meaning all living things have cells. You can't tell if this thing is living if it doesn't have a cell. Um, the cell has the ability to exhibit all properties of life. Diba? We have already discussed the 7 properties of life. And even if it's just only one tiny bit of cell, all of these properties can be exhibited by that single cell. So, what is basically the sense that we're studying the cell? Of course, if we know the structure and the function of a cell, we would have a better understanding on the growth and reproduction of all living things, how living things evolve and pass on their hereditary traits from one generation to another. And of course, their organismic functions. Like, for example, what are their roles in the ecosystem? Okay. And now let's go back to the history of the discovery of the cell. It all started when Robert Hooke was able to observe the different pangalan yan, dead cells in a cork. He was able to like cut a very thin slice of pangalan yan, a cork, and then put it on a very simple microscope. And then he observed this, ito siya. Wait lang ha, I'll put it into red. Ito siya, na mga red, na mga small rooms, and he coined it as cells or cellula, diba? So parang cells na selda ng pulungan, ganon. But Robert Hooke discovered or observed dead cells from the cork, diba? It was only until Anton van Leeuwenhoek who have, pangalan yan, observed the first na mga living organisms with the living cells. So, the first na organism that he was able to observe is the Spirogyra. This is actually an algae. In 1674, um, So, ito siya yung ano, spirogyra. So, para siyang spiral yung from one cell. It's like that. And then from then on, um, wait lang ha, I'll just erase the drawings. Yan. And then from then on, there was a convention of scientists or the great minds in those times that in order to be considered living, an organism must have or must be composed of at least a single cell. Siya nag-notes. Okay. From that discovery of the cells, especially the living ones, three different na mga prominent minds during 1800s have conceptualized the cell theory. So the cell theory is actually attributed to the botanist Matthias Clayden, the zoologist Peter Schwann, and also to Rodolphe Virchow. So actually, it was a kind of parang ano, a coincidental story in which Matthias Clayden and then Peter Schwann had a little bit of tea time and then pep talk and then they were discussing about their observations of the cells that they're interested in. So like for example, Matthias Gliden is interested in plant cells and then Theodore Schwann was interested in animal cells. And then they found out that plants and animal cells have similarities. So that's why they made general rules about the properties of the cell through the cell theory. So here are the tenets of the modern cell theory. Number one, all organisms are composed of one or more cells. So in order to be called as living, you must have at least one cell. And number two, cells are the smallest living units of all living things. Number three, cells arise only by division of an existing cell. Which is in Latin term, it is also known as omnicellula e cellula. And then Rudolf Virchow finally have further improved or like elaborated the cell theory and added four more tenets on it. And these are number four, all cells are basically the same in chemical composition and metabolic functions. Meaning, what you see in one cell, you can also see in other cells, especially in their basic composition. So, like for example, you can see the building blocks. So, like the carbohydrates, amino acids, the fatty acids, um, and then the nucleic acids there in the cell. And of course, the metabolic functions. So like for example, how it grows and then how it also reproduces and then also how it, pangalan yan, it uses energy in order to function and grow and develop. And then number five, all basic physiological functions are carried out inside the cells. So, like for example, reproduction, di ba? An organism can reproduce. And also, a cell can also reproduce through cell division, digestion, di ba? An organism like a multicellular organism can digest its food. Well, the cell can also digest, di ba? In a sense that it can break down the bonds of pangalan yan, of polymers pa. Oo. so that it can generate the energy, diba? Pangalan yan, needed for it to function well. And then, respiration. So, like for example, us, we need to respire, and yes. While in the cellular level, we also have what we call a cellular respiration, which is actually, pangalan yan, the usage of those, pangalan yan, the breaking down of those na mga ano, monomers para pangalan yan, maka-create og ATP, and then also may use up ni mga imong oxygen. Yeah. And this is a more complex na nga ano, discussion in cell and molecular biology, in the third year for the pangalan yan, old curriculum. But in your case, marag di naman po ni siya need. And then, number 6, cell activity depends on the activities of subcellular structures within the cell. Like for example, the organelles that are present in there. So like for in leaves, so it has a lot of chloroplasts. So meaning it can do photosynthesis, diba? While in liver cells, these cells have a lot of smooth endoplasmic reticulum, meaning it can easily break down those toxins or alcohol. So, or yung mga ano, pangalan yan, medyo overwhelming na mga components that comes into the body, diba? Which is basically the function of the liver. And then next also the plasma membrane, and then the nucleus. Next, number 7, the last tenet of the cell theory is the cell contains hereditary info, or the DNA passed on from one cell to cell during cell division. So all cells, meaning all living organisms, have DNAs, diba? In them, because this is basically the blueprint of life. Meaning, kung ano yung magiging fate mo when you grow up, you can pinpoint to the DNA to explain why and how. And then also, the DNA is also crucial, or like it has a role in cell division. So there are 2 types of cell division, mitosis and meiosis. So yes, we will discuss it further as we go along this lecture. Okay. So there are actually 2 types of cells. We call them as prokaryotes and also eukaryotes. So when you say a prokaryotic cell, it is and will always be a unicellular organism. When you say unicellular organism, it can function even if as a whole organism, even if it just has one cell, diba? Kaya diba in our definition of the cell theory, all of the basic physiological functions of an organism can actually be found in the cell. So the simpler kinds of organisms like the bacteria and archaea are actually unicellular. So forgive me for the typo here. Archaea is ARCHAEA. And then also, these prokaryotic cells don't actually have membrane-bound organelles. So the components of this cell is actually very simple in the sense that it doesn't have any nucleus, any mitochondria, chloroplast, whatsoever it is that is found in the eukaryotic cells. It only has, pangalan yan, ribosomes for the synthesis of proteins. And then lastly, they tend to be small and very simple. Okay, so what are the different structures or the different parts of a prokaryotic cell. So this is how, or like, pangalan yan. Wait lang ha. I'll just do a spotlight yan. This is what your typical prokaryotic cell looks like. This is a bacterium. Wait, yan. So here in the mediocentral region, this is what we actually call as the nucleoid. So unlike the nucleus, which has a distinct boundary where your DNA is enclosed in the nucleoid region, this is just the space where your DNA is actually like loosely inserted in. And then these dots, blue dots that can be found all throughout the cytoplasm is called as the ribosome. So what is the function of the ribosome? It is responsible for protein synthesis. Diba? In our previous discussion, pangalan yan, proteins are very important in living organisms in the sense that they perform diverse na functions. So that is pangalan yan, mura o role ng ribosomes. Makes the proteins that are needed in the cell. And the number 3, cell wall, pangalan yan. Although the cell wall of prokaryotic cells are actually different in composition or like, yeah, from the cell walls in plant cells, medyo pare-pareho ang inyong function, which is to provide structure and protection from the outside environment. Yes. So it is made up of peptidoglycans. While the plant cells kay cellulose. Okay. Next, number 4. So cell wall is like the outside most part here ay naa pa di capsule din sa sa prokaryotic cell nga tong unsay pangalan na nga ako gi-show din hi. So if we go from the outside to the innermost, we have capsule, which is usually for attachment para mo rin siya pilit ra bitaw or glue. And then after the capsule is the cell wall for the protection and structure. And then after the cell wall, papasok is your cell membrane. And then ang katong space na dito is yung cytoplasm. Okay. So just like any other cell, of course, the function of the cell membrane is ang pangalan is to regulate, diba, the entry and exit of molecules in and out of the cell. Next, fimbriae. What man sir ko fimbriae din eh. Yes. Fimbriae are actually thin or hair-like structures that help with the attachment of pangalan yan, of a prokaryotic cell. And then the pili, these are thin tail-like structures that assist in the movement. So if it's for attachment, it's called fimbriae, while if it's for movement, it's called pili. And then in this sample of a bacterium that I have shown you, meron pa nga siyang flagellum, which is also for pangalan yan, for movement or locomotion. Okay, now we're already done with the basic structures of the prokaryotic cell. Now let's move on to the eukaryotic cell. Just to mention it pala, when you say prokaryotic, pro means before. Karyon means nucleus, meaning before the emergence of the nucleus in the cells. Walang nucleus. While eukaryote means karyon is nucleus, U is true, meaning all these cells have true nucleus inside them. So, unlike the prokaryotic cells that are all unicellular organisms, um, eukaryotic cells can be unicellular or multicellular. So like for example, your algae or like phytoplankton, they are just unicellular. Some fungi are also unicellular like your yeast, di ba? The yeast that you put on in the bread. Eh, and then, if you go and move through the more complex na ng mga living organisms, you will have different multicellular organisms, from plants to animals. Yes. And then, of course, they have nucleus. And aside from that, they have membrane-bound organelles. Now, we can see here the different mga pangalan yan, subcellular structures that we know of when we were in high school, di ba? Yung mitochondria, chloroplast, um, rough ER, smooth ER, so on and so forth. Okay, now let's discuss one by one the different cellular organelles. So number one, nucleus. So the nucleus is a distinct, di ba, na portion here in the pangalan yan, in the eukaryotic cell because it has a nuclear membrane and then it houses the DNA in a form of chromatin, di ba? Where your genetic information is stored. And then to the centermost part of the nucleus, this one, the orange one, is the nucleolus, which is responsible for the production of your ribosomal RNA. So, pangalan, para makahimo kasi mga ribosomes, naman ay 2 RNAs there. The small and the big. So, yeah. Muna gamit sa nucleolus. And then number 3, we have the plasma membrane. So just to let you know, the diagram of a eukaryotic cell that I have shown in this slide is actually an animal cell. Why can I say that it's an animal cell? It's because it doesn't have a cell wall. Okay. So it only has a plasma membrane as serving as a gate, di ba? For the that surrounds the entire cell for the entry, for the regulation of the passage of the different molecules in and out of the cell is one. Okay. And then in this diagram that I have shown you, this is actually already a plant cell. Okay. So what makes it different from an animal cell? It's because it has a cell wall. Yan, this one. The green ones. So aside from the cell membrane, if we go outside, meron na siyang cell wall. This is a rigid structure that surrounds the cell membrane and provides the cell with its distinct shape. So, di ba, if you can still remember the laboratory or like, that pangalan niya, the shape of the onion cells and also the pangalan niyan, snake plant cells, di ba, they are very distinct na mga polygonal or rectangular. And that's because of the cell wall. Cytoskeleton is actually found in both animal and plant cells, but I have just shown you here the plant cell because in this diagram, we can see clearly the cytoskeletal components here. Ito siya yung parang mga strands. So, there are actually a lot of functions of the cytoskeleton. First is for structure, like parang skeleton ng tao, di ba? For structure. And then also for cell movement, like for example, how does the chloroplast move from one side of the cell to another when there is no sunlight in this part and then merong ditong part. So it's the cytoskeleton that does it. Dinadrug na yung chloroplasm. And also in cell divisions, how do the different chromosomes or pangalan separate from each other? This is through the cytoskeleton. There are three types of cytoskeleton: microtubules, microfilaments, and intermediate filaments. Next, the ribosomes are also found again in prokaryotic cells. So meaning all living organisms have ribosomes, which is for protein synthesis. Then mitochondria. Both plant and animal cells have mitochondria because they are responsible for the production of energy that is needed, diba? For the cell to live. So, diba? Sabi ng atong marag famous quotes of mitochondria, the powerhouse of the cell. Next, cytoplasm. So, the cytoplasm is actually just the, the space, except the nucleus inside the cell, where all your organelles are actually embedded in. And then it's filled with this gel-like substance called as the cytosol. And then, meron na naman akong ibang diagram to make it better na mas distinguishable. Especially tong rough and smooth endoplasmic reticulum. So, di ba? Near here, the nucleus, you can see this, para siyang, pangalan yan, may tigyawan, may pimples. Rough endoplasmic reticulum, which is responsible for protein transport, and then, and production. It's because what makes the rough endoplasmic reticulum rough, the ribosomes are actually attached to it. So, yan. Kaya siya parang ganun. Kung titingnan sa microscope. So, parang after gumawa ng protein, ng ribosomes, ribosomes. Pangalan yan. Sinasaksak niya doon sa rough endoplasmic reticulum because it's the rough endoplasmic reticulum that packages it for the transport of that protein to the next organelle. And then we also have smooth endoplasmic reticulum. This one, ito siya yung hindi siya ina-attachan ng any ribosome. Okay? So, if the rough endoplasmic reticulum has a role in protein production, the smooth endoplasmic reticulum, on the other hand, has a role in lipid production. Diba? Our cell membranes kasi are made up of lipids. And also for detoxification. Diba? So, sa mga mahilig sa alcohol, dapat maraming smooth endoplasmic reticulum. And then aside from that, there are also free ribosomes that can still be found all throughout the cell. These dots, these very little dots are actually free ribosomes. We also have the Golgi, whatever you call it, basta as long as you write it correctly, apparatus. So this one is responsible for protein modification and export. Because sometimes, hindi pa kompleto yung protein mo. Kailangan pang attach siya ng carbohydrate, lipid, something like that. So yes. Next, we also have the peroxisome. So, as you can see, this is the cell. And then if you can see, yung parang, meron siyang piatos, tapos naka-enclose pa sa circular na na membrane, that is the peroxisome. So its function is for lipid destruction, and it also contains oxidative enzymes, usually for neutralizing mga poisons and toxins. Next, lysosome has a role in the disposal and recycling of extracellular and intracellular macromolecules. So like for example, your mitochondria is already your mitochondrion is already degrading. So your lysosome, ito pala siya, this one. Your lysosome will be the one assigned to eat it up and break it down into smaller bits of pieces so that these pieces can still be recycled by the cell. And then if a bacterium enters into the cell, it is also the function of the lysosome to eat it up para mamatay ang bacterium. Ha? Yan naman. What is the is? Pagdali kay naa pa ko'y ano. Naghimo pa ako sa picture. O tapos,
Unsa man man pwede itang taon mo?
ala nahuman na ba ya ang ano?
Pwede na pwede na? Sige
Nahuman na ag sa register ba ya? Last na gid to nga ang gihimo ninyo nga mga anis katong like yes, nag-adto ko ni Sir Jonathan.
Sige ra ma'am. Salamat ka mo.
Sige. And then, we also have, eto siya yung mga barrel shaped. This is the cell. And then we have this pink barrel shaped na mga organelles called as the centrioles. Pero in a pair, it's called as a centrosome. Pangalan yan. This is crucial in cell division. So mamaya, ano sila gamit mo sila pull sa mga chromosomes. Then going back to the plant cell, we have chloroplasts here. Ito yung mga green na capsule-like na mga organelles for photosynthesis. Basically, that's how our food from plants are formed. Diba? Dinhinga sites. And then number 16, cilia. So this is for locomotion. So as you can see here, this is one cell, a paramecium, and then it has para siyang mga bristles para makamove ang imong paramecium. And then food vacuole. Where is the food vacuole? Here. Pero lang siyang storage na organelles. Yun lang yung function niya. And then, in plant cells, we also have the central vacuole, this one, the very big na organelle, which is filled with water and sap to maintain the pressure against the cell wall. So if your plant has lacking na na water in a sense, o na mo busy, kaning imong food vacuole ang maoy kipos. Kung daghan ra po, mo ni siya ang mudako. Mulujat ay na ang plantas. Kung gamay ra ang water sa imong central vacuole. Okay, so from all of these features and pangalan yan, structures that have been discussed, we can generally say that all living organisms that have cells in them have these four universal features. First off, they all have DNA. Second, plasma membrane. Third, cytoplasm. And lastly, ribosomes. Okay, so how are molecules being transported in and out of the cell? So we will be discussing three types of transport. First is the passive transport, second is the active transport, and lastly, bulk transport. So from the word itself, diba, we can already have an idea what passive transport means. It only says that the movement of the substances in and out of the cell doesn't really need any energy to be, pangalan yan, to be required because parang nagdahil lang siya sa concentration gradient na nangyayari in outside and inside of the cell. So depende lang kung saan yung mas marami papunta sa mas konti na part yung movement ng mga substances. While active transport requires energy, di ba? In order for you to move that substance because this substance is being transported against the concentration gradient. Meaning, kahit konti lang yung concentration ng substance na to, dito, kukunan mo pa siya tapos ipapasok mo pa siya sa area na napakarami na ng molecules na ito. Okay. So we have a very simple na demonstration in here. So let's say we have a dye or like food coloring, diba? First is naglagay ka ng food coloring na blue. And then since it is the only type of molecule that is there, diba? Mag-neutralize yan, magiging uniform yan all throughout your water. And then after that, you will just put on red food coloring in just one area, diba? Here at the leftmost part. What happens is that these red molecules from your red food coloring will disperse all throughout your medium in order to make the distribution of the red molecules uniform all throughout your water medium, diba? And then eventually, the distribution of the blue and the red molecules will, will equate. Yes. In a sense na magiging isotonic siya. Okay. So that's basically the diffusion. Di ba muna itong gimin sa diffusion? Just the spontaneous movement of a substance from one region nga higher concentration gradient to low concentration gradient. So, let's put a membrane in between those molecules. So, what does it mean by a membrane? The cell membrane. Let's imagine that this is the cell membrane. And then we have one dye, only one dye, here in the left side of the cell membrane. So in the passive transport, what the molecules naturally do is that they will transfer slowly to the area where there is less concentration gradient. In this part, ito siya na area sa right side where there are no molecules present here. So yan, magta-transpose yung mga molecules slowly from the left to the right until magiging equal na yung distribution of molecules in the left side and the right side, reaching the equilibrium. Now, what if you have two different types of molecules, diba? So, same pa rin yung, pangalan yan, yung principle. So, this purple dye will move to the right while this orange dye will move to the left until you will reach the equilibrium stage wherein you will get the same number of orange dyes in the left and the right sides and also the same number of purple dyes in the left and the right side of your membrane. So what is the importance of diffusion? Pangalan yan. In our red blood cells, di ba? Kung marami yung, for example, solutes, mga salts, kinain mo, something like that. What happens is that your solution, like your fluid, will become hypertonic. So the water molecules in your red blood cells will come out from your red blood cells, thus making it shrink. So hindi na siya nakaka-function well. And that's what we call as a hypertonic solution. Okay? While, for example, nasobrahan ka din sa pag-inom ng tubig, di ba? Nakakamatay kapag sobra sa pag-inom ng tubig. Bakit? Because it will make your vessels become hypotonic in a sense na sobrang napakarami na ng water molecules outside the red blood cells, causing the water molecules to surge into the red blood cells and eventually bursting those red blood cells. So, buba, yung red blood cells. So, what your pangalan yan? Your goal is just to achieve the isotonic solution. Di ba? Kaya nga tayo dinidextrosan kapag wala ng tubig, something like that. Para or ka nang hindi na nakakakain ng food, kaya para maka-achieve lang ng isotonic na solution in your bloods, in your circulatory system, yes, in your blood, yes. Yeah, that's it. So when you say isotonic solution, the going out and the coming in of the water in the blood is just equal or in equilibrium. What about in plants? So like for example, grabe yung drought, di ba? So, yung parang result niyan is that yung water solution, yung water in your central vacuole will come out of the cell, making it plasmolysed. And then, kapag grabe naman yung like nagbaha or something like that, because it has a cell wall, hindi siya mag-burst unlike the animal cells, like the red blood cells na mag-burst talaga siya. Kasi, your cell wall is very rigid and it will, pangalan yan, it will prevent it from happening, yung pag-burst. Diba? Kaya nga nakaka-survive ang mga plants kapag baha ng ilang araw, something like that. So, it will just make the plant cells turgid. And then, if there is just an equal movement of water molecules in and out of the plant cell, then those plant cells will just be flaccid. So, like, yan, ito siya yung forma. Kapag hypertonic yung solution, maraming solutes or yan, wala talagang water, something like that. So, pangalan yan. Mulujat. Kapag isotonic, okay ra siya. Pero kapag hypotonic, ano dyan siya? Turgid. Hindog yung duro-duro. Okay. So, osmosis again is just the movement of the water in and out of the cell. It's just a special type of diffusion wherein it focuses the movement of the water. Let's move on to active transport. Again, when we say active transport, it needs energy in order to move a substance against a concentration gradient. So just like what we can see here, pangalan yan. Yan, konti lang yung mga molecules in the inside of the cell, but yung sa outside, napakarami na yan. Pero ang response pa rin niya is to pump more and more molecules outside of the cell. So how does it do that? Na pangalan yan, medyo Kung sa friction pa kayo, grabe ang friction. Kailangan niya ng energy to pump it outside of the cell. Lastly, we have bulk transport. So bulk transport is actually assigned for the movement of large quantities of materials across the cell membrane. So we have endocytosis for the intake of large materials inside of the cell, while exocytosis is for the release of those large materials outside of the cell. So yeah, here it is, the diagram. This is exocytosis, this is endocytosis. Okay, so if you are taking in fluid in large amounts, yung cell, it's called a spinocytosis, and it's also known as cell drinking. Yan. While if the cell is taking in large amounts of solid material, it's what we call as cell eating. So like for example, how does the white blood cell kill a bacterium, diba? It needs to eat it whole. So yes, that is an example of cell eating. Yan. I have provided here some pangalan yan, some links where you can actually watch this. Let me see though, if we still have enough time. Types of endocytosis. Wait lang. How can I? Yan. I'll just activate the audio. Share computer sound. Yan, this is your amoeba. This is a parasitic na protist. And it's eating a paramecium as its food. So, para siyang finger, dalawang fingers. Until magana na siya. And then magpo-feast na. Yan. Phagocytosis.
Ang lungkot pag may sakit, pero pag magaling na ako, balik super kid na ulit!
Choose Calpol. Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
That's it. And then, let's move on to exocytosis. Yun lang, sinisecrete lang yung waste products. Like, more siyang malibang. Maura na siya. Okay, last part of our discussion about cells is the cell cycle and cell division. So, let me ask you a question. Why do cells need to divide? Bakit kailangan mag-divide ang Well, ironically, cells need to divide in order to multiply. In order to, pangalan yan, supply needed ng mga cells, especially to eukaryotic na mga organisms. In a sense, like for example, in reproduction, like your zygote needs to divide like in a lot of times, 'di ba? Para maka-form ka ng your anong pangalan niyan? Embryo and then fetus and then eventually kapag na may mga infant and then also after panganak na ang infant, it needs to grow and develop, so still need to divide para maka-keep up with the height na requirements, the weight na and then the different na mga metabolic functions of the body of a eukaryotic organism. And then if, for example, there is an accident that happens all throughout the life of that organism, like nasamad siya, cells need to divide in order to renew the tissues that are being destroyed or like destructed. Diba? So, para lang yan siyang series of division and then grow, division, grow, division of the cell. And then that's what we call as the cell cycle. But the division of the cell is divided into or like classified into two. We have mitosis and meiosis. First off, let's describe the eukaryotic cell cycle. Yung parang balik-balik lang. Grow, divide, grow, divide, grow, divide. Ayan. So, in the most part of a cell, it is actually in the interface. Meaning, it just focuses on growing. And then in the synthesis part is the replicating of the DNA. And then in the gap2 is further growing and also further production of parang murag duplicate copies of your organelles para it becomes ready for the distribution of the organelles equally when it produces two identical na mga daughter cells in mitosis. Yan. So most of the life of the cell is in the interface, which is divided into three, gap1, S phase, and gap2 phase. Ito siya. Again, gap1 is just for growing of the cell. Synthesis is the replication of the DNA or DNA synthesis. Gap2 is the further growing in order to make it ready for the cell division, which is in somatic cells, what we call as the mitotic phase or mitosis. What is a somatic cell? So when you say somatic cell, all of the cells that you have, except the germ cells. Cells na nagpo-produce ng gamits, yun yung germ cells. And then, in the mitotic phase, even if it's just a very small portion of the cell cycle, it's still divided into 4 parts. The prophase, metaphase, anaphase, and telophase. And then when it's ready, it will separate into 2 through cytokinesis. Okay, so what happens before the cell gets ready to divide? Of course, in the gap2 of the interface, the nucleus is still there. I will just point these na mga structures para makita niyo yung somehow the difference when it enters the mitotic phase. So see, as you can see here, the boundaries of the nuclear membrane are still intact, while the nucleoli or nucleolus is still present. Though parang hindi naman klaro dito. Then, your centrosomes are replicated here. Ito siya. Ito yan, 1 and 2. Then, the asters are present. 1, ito siya. Yeah. And then, of course, tapos na yung pag-duplicate ng DNA doon sa S phase, diba? Before the gap2. And your DNA is still in chromatin form, meaning they're still in loose form, hindi pa sila compact. As you can see here. So if they're in loose form, hindi actually sila klaro. Pangalan yan sa microscope. But when it enters the mitotic phase, which is the first part of it, is the prophase, your chromatins will condense and form into chromosomes. So, chromosomes na yung pangalan kapag nagkakondense. Ito na siya, parang siyang mga compact na mga yarn na naka-X, ganun. Also, your nuclear membrane will also disappear. Wala nang nucleus na distinct. Your centrosomes will also separate into the opposite poles. And then, yes. Ano to? After that, your chromosomes will, move to the center na plate, which is what we call as the metaphase plate. So, nasa gitna na siya. And then your centrosomes are here at the poles because they will be the ones responsible for pulling the identical na mga sister chromatids. In this case, upward and downwards up to the north and south poles of the cell. So, take note ha that, di ba, pangalan niya yung sabi doon sa G2 of the interface is nakaduplicate ng DNA. So, parang nakaganon siya. So, isang identical na copy, another na identical na copy. Tapos, i-orient yan siya na merong isang identical na copy na naka-face to the north and then face to the south in this na diagram. Kasi yung hihiwalayin yan sila sa anaphase. So, ayan. In the anaphase, meron ka pa rin isang copy ng yung DNA mo na mapupunta sa north and sa south of the pole. And eventually, pangalan yan, mag-divide kasi yung cell into 2 daughter cells, di ba? So, ayan, your sister chromatids, hindi na sila magiging sister kaya naghiwalay na. Magiging new chromosomes na sila. Here, one copy here, and then another copy here. And then, yan yung parang distinct na nangyayari sa anaphase, the separation of the sister chromatids. While in the telophase, those chromosomes will now reach the opposite poles and the nuclear envelope reforms, and then pangalan yan, your chromosomes will go back to being chromatids. That's why it's called decondensation. And also cytokinesis, or the pinching out of the cell in order to make 2 new identical daughter cells. Identical in a sense that it has the same set of chromosomes in each of the cells. Yes. Yan. In animal cells, what actually happens is that parang may pinching na nangyayari. And this pinching is called as the cleavage furrow. While in the plant cells, para kang naglalagay ng semento lang dito sa gitna. Para makamake ka ng 2 new daughter cells. Yan. So, what is the description of the daughter cells? They need to be 2 identical. When you say identical, genetically identical from the parent cell. Kung isa lang yung parent cell, magiging 2 pero pareho lang sila. Diba? So, parang photocopy na times 2. So, are produced and they are also diploid. So, say diploid. Kung ano yung nandoon na set, yun pa rin yung lalabas na set sa daughter cells. Okay, so let's have a review of the cell cycle and cell division. Here in the somatic cells, so of course, this one is still in interphase because as what you can see, the nuclear membrane is still present. And the chromatins, or there are still no chromosomes, di ba? Hindi pa nag-condense. So, your DNA is still in chromatin form. While this second picture is prophase, in the sense that you can see the compacting na ng mga chromosomes and the asters and centrioles moving to the opposite sides of the pole. While here in this third picture, it's now in the metaphase stage because all the chromosomes have, pangalan yan, have been moved to the center plate, which is called as the metaphase plate. And then, when the sister chromatids will be separated, that is what we call as the anaphase. And then, when it moves to the, opposite na poles of your, pangalan yan, slowly forming two cells, that is what you call as the telophase. And kapag, pangalan yan, naging complete na yung separation of the two new daughter cells, that event of separating the cells, the two new daughter cells from each other is called as cytokinesis. Then, yes, magdi-decondense na ang imong mga chromosomes. Yan. This is a simple GIF of mitosis. Yan, two new cells are formed. So prophase, interphase, prophase. And then prophase, parent prophase. And then when it reaches to the metaphase plate, metaphase, anaphase, telophase, and then cytokinesis. Okay. For the last portion of the discussion, this is the final pangalan, the second type of cell division, which is meiosis. So meiosis is the special type of cell division in germ cells. So meaning these are the cells that actively dividing cells in your testes and ovaries to produce your sperm cells and egg cells, diba? So meiosis is only found there among all the different parts of your body, diba? And then it produces gametes with one haploid set of chromosomes. Unlike in the mitosis, where you will, pangalan yan, produce diploid cells, in meiosis, you will produce haploid set of chromosomes. So like for example, we have here the parent cell. So you can see it has 1, 2, 3, 4 chromosomes. But at the end of the meiosis, each cell will just have 2, diba? 2 nga chromosomes. So from 4, naging 2. That's why it's called as haploid. Naging half na lang siya. But, pangalan yan, you don't have to worry about it because when two gametes, fuse together in fertilization, then that diploid number will just be conserved to form your new organism. So, yes. Another difference of meiosis from mitosis is that it has 2 stages. It has meiosis 1 and then meiosis 2. Here, meiosis 1 is also called as reductional division because this is where your 2 sets of chromosomes becomes half. So, naging 1 set na lang. And then in meiosis 2, it's called as equational division because, diba yung chromosomes mo dalawa, iaano din yan sila. Na parang sa mitosis na part. Okay. So, what happens in the prophase 1? Basically, parang pare-pareho lang din sa sa mitosis, mawawala yung nuclear membrane, nucleus, ganon. Pero, what is distinct here is that your homologous chromosomes pair. When you say homologous chromosomes, these are your similar shape, similar size chromosomes coming from your mother and your father, and then they will pair up because they will do crossing over. Like exchange yan sila ng mga genes or sets of genes, di ba? That is the reason why hindi pare-pareho ang mga anak ng itsura. Kasi in exchange yan sa prophase 1. Next, after prophase 1 is the metaphase 1, same pa rin na pangalan yan. Your chromosomes will align in the metaphase plate, but instead of it having only one line, so para siyang in pairs. And then, in anaphase, this is where your homologous chromosomes will separate from each other. So, as what you can see here, diba, yung isang part niyan, let's say the blue chromosome part is from your father in the features, and then the orange chromosome part is from your mother na features. So, this one chromosome here is made up of one sister chromatid. Di na din yung sister kay lahi-lahi ng pangalan. Chromatid. And a purely father features. And then the other one is mostly father and a little bit of mother features. And then, dito sa baba, yung another na homologous na chromosome is, meron siyang mostly mother and little bit of father. And then, dito sa isa niya na is mostly mother, like all of them mother na features. And yan. Next, magse-separate yan sila na dyads or groups of two non-homologous chromatids. And then, magiging haploid na ang number of chromosomes that will be distributed in each of the cells that will be produced at the end of the meiosis 1. So, there will be 2 cells produced. So, from initially 4 chromosomes, magiging 2 chromosomes each na sila. Nag-half na. Okay. And then, that's the end of meiosis 1. And then, in meiosis 2, this is just the same as mitosis. Lang sila? Kay prophase 2, metaphase 2, anaphase 2, telophase 2. Then, same lang katong nahitabo sa mitosis. Pero ang difference lang nila is that dili sila mga genetically identical. Okay. In the first place, di ba, nagka-mix and match naman tong father and mother genes. And eventually forming 4 genetically distinct cells at the end of one meiosis period. So yes, that's it. And haploid. Yan. Okay. Lastly, let's discuss the difference between spermatogenesis and oogenesis. This is spermatogenesis. It's just the production of your sperm cells in the testes. At the end of the spermatogenesis, you will produce 4 live and viable haploid sperm cells. But in the oogenesis, you will just produce 1 viable egg cell. It's because your cytoplasm is actually not divided equally. So, namamatay yung the 3 pang mga polar bonds na napoproduce. So, that's the difference. 4 and then 1. And then, pag yun nga sa spermatogenesis kay like everyday reproduce. Well, ang oogenesis kay diha na siya kapag every period lang, every month. So, yes. Here is another GIF of pangalan yan. So, this is meiosis 1, telophase 1, and then prophase 2, and then metaphase 2, anaphase 2, and then telophase 2, forming 4 haploid genetically distinct cells. So here is the summary of all the discussions that I have made. Actually, you can just put it in, in a table with these different properties, and then you will have all of those things that I have discussed. So basically, that's it. All about these cells. And then please watch and study this lesson, because in the next meeting we will have an assessment and also oral recitation, and then for the next meeting after that we will have a long quiz. Thank you for listening.